Thank you very much, and thank you for allowing me the chance to talk today. As Debbie mentioned, I am Elaine's stand-in today. She couldn't make it, but I'll try to cover the information that she was going to talk about. So in 20 minutes, I really don't have time to cover the entire field of canine cancer genomics, so I'm really going to talk about a couple of our most recent studies that have been going on in our lab and with our collaborators. So this is just a little bit of what we already heard this morning, and it looks like my numbers are more out of date. Everybody else had a little bit better numbers. But there are a lot of dogs in the United States. A lot of dogs, and as we know that they live with us, they're exposed to their same environments that we are. They receive preventative and diagnostic care, second only to human. Dogs live into old age. Even the shortest lived breeds, some that only make it to about an average of 8, are twice as long lived as their wild progenitors. So they're developing a lot of different diseases, and a lot of the diseases that we're very familiar with. And what's attracted a lot of us to the study of dog genetics is the fact that they have this population structure, which is the breed structure, which is well suited to studying the inherited forms of cancer. Cancer is the #1 cause of disease-associated death in dogs. More than 1 in 4 dogs will develop cancer over their lifetime, and that increases with the age of the dogs. And many of the breeds, as Debbie was just mentioning, many of the cancers show a strong breed predisposition. Gastric cancer, 10 to 20 times higher risk in chow chows than the average mixed breed dog. Golden retrievers have a huge risk of hemangiosarcoma. Airedales have a very strong risk of pancreatic carcinoma, and of course the Scottish Terriers down here with nearly 20 times increased risk of developing bladder cancer. I am not, however, going to talk to you today about how we are mapping inherited forms of cancer. I'm going to talk to you about some of the work that we've been doing looking at the tumors that develop in these breeds. Starting with canine transitional cell carcinoma of the bladder. So this is a study that we do in collaboration with Debbie Knapp's group over at Purdue. Transitional cell carcinoma of the bladder makes up about 2% of all tumors that are seen in the dog, and this is very similar level as bladder cancers appear in human populations as well. It affects about 20,000 dogs each year, and Scottish Terriers at a very high risk, nearly 20 times more likely to get it than a mixed breed dog. But we see other breeds at high risk as well, such as Shetland Sheepdogs, West Highland White Terriers, about 5 times increased risk, and there are a few other breeds as well that get a high incidence of bladder cancer. So while we were doing some mapping, genomic GWAS studies to look for the inherited components, we realized that we needed a little bit more genome information to understand this tumor. So we decided just to do a little survey sequencing on some of the tumors that we had. We did 4 tumors from some of the high-risk breeds, the Scottish Terrier, West Highland White Terrier, and Shetland Sheepdog, and then a couple of normal bladder tissues. And using some Illumina HiSeq, we did a complete transcriptomes on each of these tumors. The idea, of course, was to identify transcripts that might be present in these tumors that weren't present in a normal dog genome, to find genes that were only expressed in the tumors. And then finally, we decided to look at this sequence to identify somatic mutations in these tumors. And what we found was in canine bladder cancers, there's a high incidence of a mutation in the BRAF gene that is identical to the BRAF V600E gene that is found in a large number of human tumors. So this mutation in the dog right now, it's called BRAF V595E. I believe the dog is missing, and this may just be an annotation issue right now, a couple of the first codons of this protein. But it is identical to the location of V600E in humans, which has been found over 30,000 times, and in 6 or 8 different cancer types. What's interesting is it isn't often found in human bladder cancers. It's only about 1 or 2% of human bladder cancers that actually see this mutation. We did some functional studies to look at whether or not this mutation was actually behaving the way the human V600E mutation is. And we see where we have a dog cell line here that does not have the BRAF mutation versus 3 that do, that phosphorylation of MEK is increased tremendously in the individuals that had the activating BRAF mutation. So it is doing the same thing as it's doing in the human tumors. treating these cell lines with vemurafenib, which is one of the inhibitors that targets specifically V600E, we see some variation in the way that it responds. Some tumors respond well with a reduction in the phosphorylated MEK. However, other tumors don't respond nearly as much. And of course, we see very little response at all when we're looking at the tumors that don't have the BRAF V600E mutation. We also see this similar trend when we're looking at the growth of these cells. We see significant reduction in growth in a couple of the tumor cell lines that have the mutation. One has a reduction in growth, but it's not nearly as much. And then, of course, the other one responds very little because it doesn't have the mutation. So this gives us that opportunity to start looking at how targeting this particular mutation, so these personalized medicines that we've been talking about, are working in this particular tumor. It's clearly not responding the way a melanoma, a human melanoma would respond, but it is responding in the range of a colorectal cancer or perhaps a thyroid tumor would respond to these similar inhibitors, giving us a chance to look at, again, combining drugs and how these drugs will react in different tumors by looking at this naturally occurring tumor in the dog. By doing amplified resequencing of this particular tumor spot, we're able to detect it in a little less than 1 in 1,000 reads. So we can actually find this mutation in free catch urine from the dogs. And this, of course, is a benefit to the dog treatments because they'll be able to use this information to help diagnose this and perhaps go into new treatment strategies when we start looking at using inhibitors in these dogs. And of course, this also brings up the question, why do some of the tumors, I didn't mention actually, that we actually found this tumor in 85% of the canine bladder cancers that we looked at. And I believe we looked at something in the range of 65, 67 different tumors. And that's obviously a very high number that carry this one identical mutation. So what we started looking at now, and we have currently underway, of course, is looking at the transcriptome of more tumors. The first one was just a survey sequence. Now we've sequenced 22 different tumors and additional normal samples so that we can look at the differences between not only tumors and normals, but to look at the differences between the tumors that carry the BRAF mutation and those that don't, and what other mutations they may be carrying that are affecting how they respond to treatment. So I'm going to switch gears just a little bit and talk about another canine tumor. And this is canine transmissible venereal tumor. This is something that is a rather rare thing in general, but it shows up in dogs and a few other species. And what this is a tumor that developed in one individual and is actually passed by passing cells to other individuals. So this tumor has lived for quite a long time. It's a clonally transmissible parasite because it is passed from one dog to the next. It is the world's oldest known continuously propagating cell line, basically, because it propagates within the dog itself. It's transmitted during months of long periods of evasion of the immune system. And all of them have a shared origin. So it affects dogs all over the world. But so far, all information about the genome of this tumor suggests that it all comes from a single dog. So what can this tumor tell us about how a tumor can persevere within the individual? Well, let's start with the founder. It's been estimated possibly 10,000 years ago, a tumor developed in a dog. This has been passed on for generations and generations, and each time it's transmitted to another dog, it has to go through another round of immune surveillance in order to live. We can see that there are many lines that I'm sure developed mutations or didn't develop mutations that then were lost over the years, but down through the thousands of years, we've come up with the tumor today that seems to have been able to resist all of those immune steps. We have looked at the sequence that was done by Liz Murchison's group at Cambridge of 2 different CTVT tumors, one from a purebred dog in Brazil and one from possibly a mixed breed in Australia. And then looking at the most common ancestor of those 2 tumors, looking at what those changes are that happened within the tumor as it was developing. So if you consider looking at the sequence of a single tumor such as this, what we see when we look at all of the mutations are, of course, that there are mutations that come from the host dog, some contamination there. You see mutations that have happened over thousands of years of development of this particular line of this tumor. You see the somatic mutations that lead to the development of this tumor. And you see mutations that are inherited from that original dog that the tumor came from. And of course, when we're looking at any kind of a sequencing system, there are some kind of systematic errors that are involved. So by combining the tumors from two different dogs from different parts of the world, we start to cut out a few of these mutations and narrow in on really the mutations that happen within this tumor. So as you can see, we're taking out the host contamination because that's going to look different in the two different dogs. And we're taking out some of the lineage-specific mutations, so things that have developed through drift over years of propagating this tumor. And what we're left with are the somatic mutations that cause the cancer or let the cancer continue to grow and live, and then inherited alleles that come from the original founder. So what we really need to do is figure out how we can separate these two so we can understand which mutations are really propagating this tumor. What we have developed in our lab is a large catalog of variation from the dog genome. So we have been doing some sequencing of whole genomes from lots of different breeds in our lab, probably about 50 thus far, but we've also been collecting all of the sequencing that's being done in the community. And this is showing a summary of about 186 breeds, and I believe we have up to 380 different dog and a few wild canid sequences now that we've put together, covering close to 20 million SNPs. Over 20 million, actually. There's 28 million right there. So what we're looking at here is the individual dogs and the amount of variation that is covered by this catalog that we have put together. And what we estimate is that it's over 99% of the variation in single nucleotide variants and over 95% of the variation in structural variants. And this is by comparing what we find in the catalog to each of the individual dogs and how many times we can see it repeated. So if we take this catalog of variation then and apply it to our tumor, we can start to separate things that are germline variants from things that have developed somatically. This is what the experiment looks like. We start with the founder and the tumor that he was carrying, and then look at these two dogs that represent the modern lines of that tumor and all of the variants that were found within it, which are about 12 million. When we do that comparison and look at only the things that are shared by the two tumors, we next compare that to the catalog of all the variants that we have found in the dogs so far. If they're found in that catalog, then we assume that those are things that are inherited from the normal founder. These are germline mutations. If they're not found in that catalog, these are our putative somatic mutations and the ones that created this tumor to begin with. This takes us from 12 million mutations and variants down to about 1 million in the final catalog of total somatic mutations in this. Now remember, 1 million is a very large number, but this is a tumor that has been growing for possibly 10,000 years, so it's had a lot of time to build up mutations that are going to help it grow and continue. So if we start looking at where these mutations are, this tells us how this tumor can begin to live for this long and continue to propagate itself. We start out with 57 different genes that affect ubiquitination that have been knocked out in this tumor. Then we move on, 14 genes that affect the proteasome, and all of the genes that affect MHC1 loading have been knocked out in this tumor. Right up to the MHC1 genes themselves, we see mutations that prevent this gene from being able to present an antigen. So right there, we're starting to see it is blocking the ability for the immune system to find this. If we look at the entire, not the entire, this is obviously not everything, but a large amount of the genes and the pathways that are required for immune surveillance, we not only see the MHC1 genes starting to be knocked out, but all of the genes in this pathway, we have seen mutations that prevent them from being able to work and to present antigens and to allow the immune system to detect this cancer. In that way, what we're seeing is this evolutionary pressures over thousands of years find us finally developing these functionally overlapping mutations to evade host immunosurveillance. So this gene has perfectly presented itself, this tumor, to live in multiple dog breeds and to propagate itself. When we looked at the age of some of these mutations, and we did this by comparing allele frequencies within the host and within the somatic mutations, we can see that some of the earliest mutations seem to have been in very commonly affected cancer genes. We see tumor suppressors, so cell cycle control genes, and of course those that affect MHC class 1 presentation. These LOH events are some of the oldest and definitely lead into those very first generations of the tumor and what was required to begin this process. This is a look at the karyotype of a couple of these CTVT tumors. The normal dog is on top, and these are two different CTV tumors on the bottom, one from Cape Verde and one from Italy. In many cases, the structural rearrangements, like you see here on what is sort of chromosome 3, are almost identical between the tumors. There are things that have taken place and have been retained over these many, many years. But in other cases, for instance, at chromosome 1, you see very different structural variations, and we don't know what the meanings of the different structural variations are compared to the ones that are retained. We have actually identified more than 7,000 different structural variations. This particular graph is showing about 1,000 of the ones that only go from chromosome to chromosome. They're not showing the interchromosomal rearrangements that take place. 2,000 of them disrupt at least one exon in a gene, and there's many gene fusions that have shown up. And many of the mutations that we see, the single nucleotide variants and knockout mutations, may contribute to this overall instability of the genome. But because of this large number of structural variants, what we've actually decided to do is to do a de novo assembly of the CTVT tumor, sort of looking at it as though it is a new species rather than simply a comparison of the dog. We have just recently obtained fresh tissues from a new sample that was collected down in Florida from a husky, and it is actually at NISC right now. We'll be undergoing about 150x coverage with Illumina, then a PacBio long reads to do a backbone, so that we'll be able to actually put this together and annotate the tumor, the cancer itself, and look at the genes that are required to continue to grow, the genes that have been removed, and those that have been mutated to make sure that this cancer has a very long life. So the conclusions for CTVT. Basically, our canid variation catalog enables us to separate germline from somatic mutations. This catalog was also used when we're looking at the RNA-seq from the tumors that were collected in the TCC samples. This tumor, CTVT, is exquisitely adapted to its role as a transmissible allograft. It has lost almost all ability to present antigens to the new host. The early somatic mutations point to genes that may have contributed to establishing this transmissibility at the very beginning. And understanding this biology can very well shed light onto the host-tumor interactions that we see in current cancers, both in dogs and in people. Of course, we have the sequencing underway to develop a tumor genome of the CTVT. So overall, I think what we're looking at when we're talking about dogs and cancer and genetics is that dogs acquire multiple forms of cancer, and through natural means. Aging, interaction with the environment, and of course, inheritance is involved. The analysis of the TCC mutations reveals a driver that's identical to what we see in humans, but it's in an unexpected place. It's not in the cancer that we expect to see it in. This makes us think that perhaps we should start looking again at the molecular background of these tumors and how they compare rather than only looking at the cell types or the presentation. And then finally, the tumor parasite, CTVT, provides us data regarding how cancers can escape immune detection, which of course is important when we're thinking about how we're treating them and how the body, we can use the body's own mechanisms to fight cancer. As I mentioned, this is not only our lab, but this is also work from Debbie Knapp's lab at Purdue Comparative Oncology, and Jim Mulliken at NISC is working with us on the new CTVT tumor sequence, and I would be happy to answer any questions. Thank you. Am I beyond?
I think we're going to take questions at the end after all four speakers.
Okay.